I think chapter 3 is probably the most challenging. And I have this little intro slide, how do we generate a whole bunch of different molecules? Like we started with individual protons, electrons, and then we ended up with elements, hydrogen, oxygen, nitrogen. And now we're gonna put all those, the hydrogen, oxygen, nitrogen, those 4 elements together to make big molecules, to make large molecules, to make all the molecules that make up our body. Are you with me? All right, so let's start doing that. So that's what this is all about. That's what this chapter is about. So, in actuality, we're kind of like building a molecule. We're building an organism. We start in the very beginning with subatomic particles, and then we go to the elements. Subatomic particles mean protons, electrons, and neutrons. And then we go to the elements, and we're taking those individual elements, are the carbon, oxygen, hydrogen, nitrogen. We bond those together, and we make the big stuff in your body, like the carbs, the carbohydrates. Are you with me? So we're actually going to the point where we're building blocks that are in your body. Okay? That's what this chapter is about. It's about building how do we build these large molecules? We call them macromolecules, macro meaning big. And poly also means big. We're making big molecules, and we're trying to explain how we're making these big molecules that make up your body. The predominant ones are, you've heard all of these. The predominant ones are the carbs, the protein, the nucleic acid, the lipids. These are the major. There's some other ones, but now I'm going to ask you a question, because I kept saying it over and over and over in class. I said, because let's see, carbohydrates are a bunch of carbons linked to a bunch of hydrogens, and then some oxygens. Okay? That's what carbohydrates are. Because we said carbon, oxygen, hydrogen, nitrogen. So it has everything except the nitrogen. And I kept saying in class, how do I link, by link, I mean, how do I link a whole bunch of carbons to a whole bunch of hydrogens, a whole bunch of oxygens to make this? What's the method for linking those things together? What do I have to remove between each one of them? Dehydration. Remember I said that for all of these mechanisms, if I want to link a whole bunch of these together to make a huge carbohydrate, like here, in this next slide right here. So let me go to, okay? So this is actually a fatty acid. This is another fatty acid. But if I'm linking this carbon, let me get another color actually. I'm linking this carbon, that's too big. Okay? If I'm linking this carbon to this carbon to this carbon, because that's how I make a molecule. That's how I generate a molecule in my body. Okay? If I link all of these, in order to link one carbon to the next carbon, somebody's going to give up an oxygen and somebody's going to give up H2O, 2 hydrogens. Are you with me? H2O. Are you with me? Yes? So I'll show you how that works. That's very important, okay? So now, and that's the same for all of these. For proteins, I have one amino acid. I have one amino acid linked to another amino acid, links it to another one, and then somebody gives up a hydrogen, 2 hydrogens, and somebody else gives up an oxygen, which is H2O. Are you with me on that? Mm-hmm. So for all, no matter what the large molecule is, whether or not it's a lipid or DNA or carbohydrate or protein, it's made by dehydration step. So keep that in mind, a dehydration step. Okay, now let's talk about some of these functional groups first. I mentioned to you that of these functional groups, the only one I really wanted, gosh, you keep going back to the thick. Only one I really, hold on. Only one I really wanted you to know was, let me go back to my thing, and then go back here. I want you to know these two right here. Okay, I want you to, now, I want you to know what the carboxyl group looks like, and this is the acetic, and the amino group. And that's what makes proteins. That makes amino acids. Amino acids make proteins. That's why I want you to know these two. You know that proteins are made up of amino acids. Okay? Proteins are made up of amino acids. And this is the amino group, and this is the carboxyl group. And the carboxyl group is acetic. If I go back to my fatty acid, and I look at this group right here, that's the carboxyl group. That's what makes this a fatty acid. Acid. Are you with me on that? It makes it the acid. Okay? This is a fatty acid, so that group is what makes it a fatty acid. This is a fat, and here's the acid. This is the fat, that's the acid. So, and that's the fatty acid. So let's, now, if you want me to slow down a little bit on these things, I will. I do want you to know the basic part about, what the basic, I think you already know this. Carbs are for energy. Carbs are for energy. Nucleic acid is for DNA. Nucleic acid is for DNA. Proteins, proteins are for everything. And the lipid category is everything in the lipid category is hydrophobic. Everything in the lipid category is hydrophobic. What does hydrophobic mean?
Fear of water.
Water hating. Phobic is hating, philic is loving. Hydro, water, phobic, okay? So that. Now, so let's talk about how to, now I'm gonna just tell you straight up the structure. I want you to know, and I'm gonna give it to you. I'm gonna make it easy. I'm gonna make your life easy though. I'm gonna give you three structures. I'm gonna give you a structure for, a carb. If you see something like this is the glucose structure right here. Okay, this is the structure for glucose right here. You don't have to know it, but I want you to understand and carbohydrates have carbon, a bunch of hydrogens, and oxygen. Okay, now, if I give you, I might make your life easy and just, give you and say, okay, which one is a protein? Or which one is a lipid? Or which one is DNA? Something like that. Are y'all good with that? Because you're gonna see it.
DNA,
Nucleotides. Okay? So just know the overall structure. And I'm gonna give you something that's so blatantly, I'll just, since y'all are here, I'll just tell you. Usually I give a fact. I'll give a fact. Because facts look like, let me show you what fat looks like. And everybody knows what a fat looks like. So it looks like this. Okay, it's got all this stuff. Okay, so it's got this. This is a fat. Okay, it's got this structure right here. And guess how it's made? Somebody gives up an oxygen, and somebody gives up, ultimately it's H2O. to link this group right here to this group right here, and this group right here, and this group right here. They call that a dehydration step. D, without hydration, water. So without water, water is lost. So fat, I usually like to give a fat molecule, because fat is distinctly different from everybody else. It looks like a hydrocarbon because it has all of these hydrogens and carbons, but it's hydrocarbons and linked together like this and like that. Okay, yes.
Could you repeat that?
About which part?
About the fats.
Oh, well, the fats, if you look at the structure, let me kind of erase some of this stuff. This is the structure of a fat. Okay, go to color, I'll put it right green. Here's the structure of a fat. It has all of this, and this is a fatty acid portion, fatty acid portion, fatty acid portion, and then it's linked on the side by this molecule that's called a glycerol. Okay, so it looks distinctly different from everybody. So it's easy to tell the difference between that and I'm going to use DNA. And proteins look like that. They're made up of a lot of amino acids. These are amino acids. The amino acids have an amino group. They have this amino group right here and this acid group right there. And that makes a protein. Are you with me? So a bunch of these linked together, a bunch of those linked together are gonna look very different from a fat. You see what I mean? Mm-hmm. It's gonna be, and then if I have DNA is gonna look like that. Do you see the point? So it's gonna be distinctly different. Do you see how you can tell the difference between the DNA, protein, and the fat?
So are you going to give us like the...
I'm going to give you a figure. I'm going to give you an actual figure.
Okay. And we're supposed to tell the difference between the DNA, the protein, and...
Yep. And you just tell me which one it is. Okay? Okay? All right. Now let's go back and start talking about some of these. All right. Now, overall, I want you to understand. So here's a protein right here. This is a protein. These are the different proteins are made. We'll talk about later how they're made, but these are amino acids. It's got the amino group, NH2, and the carboxy group to make it acid, amino acid. So each one of these are added together via dehydration, taking water away. You take water away, and H2O to link each one of these together. And let me show you how that happens. This is a generic figure that is applicable to all the molecules that we're talking about. And regardless of whether or not it's a protein, a nucleic acid, a lipid, or whatever, a carbohydrate. To link, like for instance, forget about this one. Say you're linking this one to link this one. Because we're making these polymers. When making polymers, that's the term that your book uses. Poly just means a lot, many. Adding millions of these together to make a fat or whatever, okay? But when you're generating these, you have to take water out. That's what this is meaning. So somebody is releasing a hydrogen between, this one is releasing a hydrogen, this one's releasing an OH. So it's H2O. It's a water molecule. It doesn't matter which one, but it's a water molecule. Are you with me? That's the linkage. That's how in your body the large molecules are made. And they call them polymers. For sugar, it's the polysaccharide, polysaccharide meaning sugar. But it's water taken out. And if I'm making something that's a million, no, I'll say 1,000. 1,000 let's say nucleotides or 1,000 amino acids to make a big protein, I'm still gonna do that individually. Water taken out between each linkage. Are you with me? Between each. Water taken out. And all this says, and I'm not gonna ask you this until we start talking about how you digest your food. And then I'll start talking about adding water back, hydrolysis. But let's just focus on dehydration, and some texts will call it condensation. It's the same. So I told you in class, I said for a test, I'll put, depending on who's studying where, I'll put dehydration / condensation. So that, okay? So that will be an option. And you'll be able to, you will have to tell me what the method is. It is in order to build these large molecules, regardless of whether or not, and I might be a little tricky and say, okay, to build how do you, join together individual amino acids, and then I have a list of options, multiple guess options. And then on the next page, I'll say, how do you join together individual nucleotides to make DNA in the same option? So, and it's the same answer, dehydration, okay, dehydration, taking D meaning without, hydration, water, so you're taking water out, okay, yeah, and we'll talk about this, and this is what happens when, okay, so for carbohydrates, the individual, for carbs, for carbohydrates, the most abundant the monomer, they call it the monomer, which is the individual unit, and that's glucose. There's some other ones, there's glucose, there's fructose, there's galactose. Everybody, all you hear about is glucose, right? So it's the most common one. It's the most common, and this is the glucose. So it's got carbon, here's the formula, carbon, hydrogen, and oxygen. It's got an incredible number of all of those, actually. Okay, and the long one is called the individual one is called a monomer. Mono just means one. Polysaccharide. Poly means a lot, many. M-A-N-Y, many polysaccharides. That's glucose, that's starch, and that brings me to the difference between starch and cellulose. I want you to know the difference between starch and cellulose. And this is how they look, actually. I'll talk about this. I'll talk about this right here. This right here. Alpha versus beta when we get to starch versus cellulose. Okay? Okay. All right. Now, di means two, poly means many. Now, here's what I do want you to know. This linkage, the type of linkage. The linkage is called glycosidic. For the type of linkage for carbs, carbohydrates, is glycosidic. The type of linkage for protein, peptide. The type of linkage for DNA is phosphodiester. Okay? The type of linkage for a lipid, I didn't talk too much about it, but it's called an ester linkage. Okay? I'll repeat it. The linkage, by linkage I mean what's joining the individual units together. This link, the individual units to make a long molecule. This is called a glycosidic. Glyco means, they got a lot of names for sugar. Glycosaccharide. Let's see what else. Yeah, glycosaccharide. Yeah, but so glyco means sugar, saccharide means sugar. So this linkage, by linkage I mean joining these two together to make a long, because glycogen, which is what your body makes when you for the food, when you eat your food and you break it up, your body is gonna put that glucose back together so they can store it, store, in your liver as an energy source for you to preserve it, for when you need it. So by the linkage, I mean what joining these two together, because glycogen means millions of these together via dehydration step, just like we talked about before. But this is called a glycosidic linkage between here and here, okay? It's a covalent linkage, but it's also called glycosidic, okay? So if I'm joining individual amino acids together, it's called a peptide. So P for protein, peptide. And for DNA, joining all my nucleotides together to make my DNA, it's called phosphodiester linkage.
Can you spell it?
Oh, no. Don't even ask me to spell it. It's, let me get my thickness right here. It's called, let me go to here, let me get my, oh, this is not a good color. Oh, make it a little thinner. Okay, it's called phospho. Okay, that's an R. Phosphodiester. Okay? PHOSPHODIESTER. Okay? Okay, now, are we good on that? All right, now, let's talk about, this is a schematic of cellulose. It's got all these glucose. Millions of glucose molecules linked together to make a cellulose. Each one of those little figures, that's a glucose. So cellulose, I do want you to know the difference between cellulose and starch. And I told you that the only difference between cellulose and starch is the orientation of this linkage right here. For starch, it's an alpha. Alpha just means in this configuration, going downward, cellulose, upward. Are you with me? And that has to do with where the electrons are when this bonding interaction happens. But all you have to know is the difference between cellulose and starch is this. They have the same, both equal the same kind of linkage. They got the same kind of linkage except for the oxygen. It's in the alpha position. The oxygen is in the beta position right there. And I mentioned to you that is the reason your body cannot process this, which is the reason you cannot digest grass. You can go out and eat an oak tree as much as you want, go out and eat as much grass as you want. That grass on the field down there, it's only just gonna come out in your poop as grass. It's not gonna be absorbed and digested. And that's because you cannot process this. Cows and termites can, you cannot. But you can process this, because we eat starch all the time. That corn you were talking about, okay? Are you with me on that? This is why, this simple difference right there is why you cannot eat an oak tree and digest it. Because it's made of cellulose. It's in the wall, it's in the plant wall. It's in the plant. It's in the cell, the wall of the plant, the around the cell. And you can't digest it. So I want you to know that. It's because of this linkage. Because of this linkage, you cannot. Okay. But cows can, and termites can, because termites eat wood all day. and process it very nicely. Okay, now this is, here's where it talks about the difference between DNA and RNA, and here's the structure right here. Here's the characteristics associated with DNA. DNA, DNA compared to RNA, single strand, RNA is single strand, it's got one strand, DNA is a double helix, it's 2 strands. Now, here is where we talk about, this is where we talk about the difference between RNA and DNA, right here. At this position that we talked about at the very beginning. DNA has just a hydrogen, it's called deoxy. That's where it gets its name. D without oxy. D means without oxy, deoxyribonucleic acid. DNA is without an oxy. It just has the hydrogen. That's, to me, that's the most important difference between DNA and RNA. It's in its name. RNA has an OH right here. DNA has an H right there. Okay? That's why they call it D meaning without oxy, deoxyribonucleic acid. And RNA is ribonucleic acid. So that's the major difference between that I can see. Now, RNA also has uracil. Okay? But, and single-stranded. It's uracil and single-stranded. Making your head hurt?
Can you spell it? What's the last word?
Uracil. U-R-A-C-I-L.
RNA doesn't have that.
RNA has it.
The DNA doesn't.
Yeah. Now, if you were to see, and I'm just gonna go here right quick. So this is single-stranded RNA, and it still has adenine, guanine, cytosine, but the thymine is replaced with uracil. Okay? So here's the U. Anytime you see uracil, you know you're dealing with RNA. But you also, yes.
This is slideshow from 1 through 3, right?
Say it again.
Chapters 1 through 3? Is it a slideshow from chapter 1 through 3, or is it just one chapter?
We've already gone over 1 and 2. Okay? So, yeah, we've gone over 1 and 2, and we're gonna end with 3. Is that what you were asking? Yeah. Okay, all right, so, so let's, now 1, and I'll go over that real quick, once we finish with this, because it's not gonna be a whole lot from 1, and I could just point out exactly what's gonna be on there from 1, okay? Let's see, DNA is 2 strands, that's 1 strand, another strand. 1 strand, another strand. It's 2 strands. Now, I'm gonna get a little bit pickier towards the end of the semester when we start talking about DNA, transcription, translation, but right now I just want you to understand the fundamentals between DNA and RNA. Okay? All right. We're not gonna get too picky. It is single-stranded. RNA has uracil. RNA, and what I consider the most important, RNA has an OH on that number 2 position. DNA has a hydrogen only. That's how it gets its name. Okay. All right. Now there is this linkage between. I have to keep doing this thing right here. This linkage between on this individual strands right here. This is 1 strand. This linkage between this nucleotide and this nucleotide on the same strand. So this has got adenine, cytosine, and thymine. So here's thymine, here's cytosine, and here's adenine. So on that strand, you see where I'm coloring? On that strand, that linkage is called a phosphodiester linkage. That's the phosphodiester linkage. It's the linkage between, I can just put it on here. So here's 1 strand. This is a better example right here because it's just 1 strand. The linkage between this uracil and that adenine that adenine and that cytosine and that guanine, they are linked together by what we call a phosphodiester linkage. That's the linkage that we're talking about. And I don't have an example on here, but that's the water. That's the linkage where water is removed between somebody giving up OH and hydrogen, H2O. Are you with me on that? Dehydration. So it's the linkage between these two on the same strand. You can see it right here between the adenine and the uracil right here. You can see there's a linkage right here. That's called the phosphodiester linkage. And that makes a long strand. You can make a long strand. It can be very long. And it's the same for RNA and for DNA. The same phosphodiester linkage, actually. Okay. And I just say here it's, these three, but here you can see it better here, because you can see uracil right here linked to adenine, linked to cytosine, linked to guanine, and it's linked along here. And you see it very nicely here between the adenine and the uracil. So this is the phosphodiester linkage right here. Okay? And it is a dehydration step. It is a dehydration step. Okay, di means two. Di means two. Forget it. We'll talk about that one later. Proteins, a whole bunch of stuff. For the proteins, it's got an amino group. Proteins are